Our second speaker is Mohamed Abdel Mohsen. Mohamed was trained in the school of Ain Shams in El Cairo, and from there he moved for a postdoc in San Francisco, where he spent like a couple of years, more or less. And from San Francisco, he moved to Philadelphia, working at the Western Institute, where he spent more than 7 years, and actually where we basically met some time ago. Recently, about a year ago, he moved to the Northwestern University in Chicago, where he is now Associate Professor of Feinberg Medical School. Mohamed works in translational virology and glycoimmunology to understand how host glycans regulate immunological signals during viral infections. Most of us, we think about proteins, DNA, lipids, but glycans are always there. The glycans are decorating proteins, glycans are decorating lipids. Glycans are very important in monoclonal antibodies as well. So glycans play a really important role in biology that we frequently forget about it. So we have today Mohamed to tell us a little bit more about how glycans regulate HIV infection. Thank you.
Thank you, Javier, and good evening, everyone. And I would like to start by thanking the organizers for inviting me to be part of this exciting event, and I'm very much looking forward for the next couple of days. And I'm excited to be here with you today and share with you some of the work we're doing in our lab at Northwestern. And as Javier mentioned, our lab focusing in this emerging field of glycoimmunology, how glycans, sugars, can modulate immune responses during diseases. But today, I want to focus in one particular story, one particular project in our lab, which focuses in biomarker of viral rebound. And as all of you know, in the vast majority of people living with HIV, when they stop antiretroviral therapy, the virus would rebound very quickly within a couple of weeks, 2 to 4 weeks. However, this time vary, and there is a rare population of individuals, termed post-treatment controllers, or PTC, who can maintain viral remission for several months, sometimes years, after stopping therapy, which is obviously a very clinically relevant and desirable phenotype. However, we don't know how they do what they do. And the goal of this project was to identify host factors that can be measured in the blood of people living with HIV immediately before stopping antiretroviral therapy, and can predict how long will it take for the virus to rebound, and whether a person is more or less likely to achieve a controller phenotype. And obviously, this is important for at least 2 reasons. One, these can be used as a biomarker in HIV clinical trials, which can facilitate the success of these trials. But even more importantly, and the reason why we are doing this is we try to identify novel host pressures on HIV reservoirs during or after treatment interruption. So hopefully we can use that to develop novel tools that can enhance these pressures. And in order for us to achieve this goal, we spent some time collecting samples from a relatively large number of people living with HIV, 246 people living with HIV who underwent analytical treatment interruption in 15 different clinical trials that were conducted in 5 different countries, as you can see here. And when these people stopped antiretroviral therapy, they experienced a wide ranges of time to viral rebound. Here I'm showing you an example of how many weeks did it take to reach a viral load of 1,000 copies per ml, which happened with a median of 4 weeks. As you can see, some individuals reach it that way before the 4 weeks, but some individuals also reach it that way after this 4 weeks. Actually, some of them go for 2 years. In subset of these 246 people, exactly 74 people, we do have a very detailed viral load data that goes for many years after stopping therapy, so we can identify which of those individuals meet the criteria of post-treatment controllers, here in blue, versus ones who did not, or non-controllers, here in red. And in this study, post-treatment controllers were identified as people who can maintain viral remission for at least 24 weeks. Some of them go for much longer, but at least 24 weeks, 6 months. And I told you that the median is 4 weeks, so 24 weeks is quite extreme. And this cohort was quite diverse. And as you can imagine, most of those participants were male, but we were happy that by collecting sample from 15 different clinical trials, we were able to collect decent number of samples from female, from women. And that's actually ended to be quite important because biological sex was by far the most important or significant clinical or demographic factor we had in hand that correlated with time and probability of viral rebound. And it goes like that. Being a female was associated with 4.3 higher odds ratio. It's a lot of odds ratio of achieving a controller phenotype after stopping therapy compared to a male. And this was significant in a multivariable model, adjusting for several clinical and demographic confounder we had in this cohort, like age, ethnicity, ART initiation timing, whether they were start antiretroviral therapy at the early or late stage of HIV infection, ART duration, how many years they were on ART, and CD4 counts. Also, we had a quite different range of ethnicity, and interestingly, being non-white was associated with a slightly higher hazard ratio for a faster time to viral rebound. And again, this was approaching significance in the same multivariable model, adjusting for the key demographic and clinical factors. So I think sex and perhaps gender, as well as ethnicity, is something very important to keep in mind when thinking about viral rebound. So what we do is this sample. We focus in plasma sample collected from all of those 246 individuals, and this is what's collected immediately before stopping antiretroviral therapy. So they're still on ART. Usually this sample collected at the same time they stop antiretroviral therapy or a few days before. And we take this plasma sample, and we did over the last few years a multi-omic investigation in these samples. We did a full proteomic analysis, both untargeted using mass spec, but also targeted using a technology called NeoLISA-seq. We did a full metabolomic and lipidomic analysis using mass spec, measuring thousands and thousands of metabolites and lipids. And last but not least, we did obviously a full glycomic analysis because that is the focus in our lab. And as you can imagine, this is quite rich data set that is we keep learning more and more about viral rebound and using it as we analyzing it as we speak. And it's keep feeding different projects in our lab. But today, for the time and simplicity, I want to just focus in one of these omics, which is glycomics. And I want to give you two quick and simple examples in how we can indeed, using this omics data, identify potential candidate for biomarker of viral rebound, but even more importantly than that, how these can give us insight into immune dysfunction during and after treatment interruption, and how we can use this information in order to develop novel tools that can overcome these dysfunction. So these two examples I want to give you, one on circulating glycans and circulating glycoproteins. And as a first example in circulating glycan, the second one obviously on cell surface glycans on the cell surface of HIV-infected cells. And I will start with the circulating glycan example, and I just want to spend a quick minute telling you why we even thought that glycan, sugars, and circulating glycoproteins could be a good candidate for a biomarker of viral rebound. And in the last few years, there are many studies, actually many of them done here in Europe, that show that glycan, sugars, in circulating glycoproteins, especially antibodies, immunoglobulin, IgG, can initiate a very potent anti- or pro-inflammatory response, depending on the glycan structure. And they can also be used as a very good biomarker for the biological age of our body, in opposed to its chronological age. So I give you an example of that, of this sugar here called galactose, represented by a yellow circle. So galactose is anti-inflammatory. When a glycoprotein carries galactose, it can give it an anti-inflammatory properties that are important to protect our bodies from excessive inflammation. And as the glycoprotein lose galactose, going from right here to left, it loses this anti-inflammatory properties and become more pro-inflammatory. So it ended that as we're getting older, all of us, men, women, living with HIV or not, we systemically lose anti-inflammatory glycans, and we accumulate those glycan structure that without those anti-inflammatory glycans, which, as I mentioned, they are pro-inflammatory. So again, I give you an example here in this graph. In the y-axis, you can see this glycan structure, and you can notice it does not have this yellow circle, which means it does not have galactose, or it's agalactosylated or non-galactosylated, which put it into the most pro-inflammatory side of this spectrum. And in the x-axis, you can see age going from 18 to 100 of those men and women in this study was not living with HIV. And you can see, as we're getting older, we accumulate these pro-inflammatory, pro-aging glycans. It's interestingly, men and women accumulate this differently. Men usually accumulate this pro-inflammatory, pro-aging glycan in a very steady base from age 18 to 100, while women have this very interesting biphasic accumulation, accumulating it very slowly at the beginning until specific point, which is menopause. And then at menopause, there is a very rapid acceleration in the accumulation of this pro-inflammatory glycan to reach the same level of men or even surpass it. So we work in the field of HIV as you, and we know that people living with HIV despite long-term therapy are still experiencing a high level of many aging-associated diseases like neurological impairments, cardiovascular diseases, cancer. So a few years ago, we asked the question, does living with HIV, despite long-term therapy, is associated with an acceleration in the degradation of these anti-inflammatory glycan, or also accumulation of the pro-inflammatory glycan? So, taking a long story short, because this was published last year in this paper in Nature Communication led by Leila Giron in our lab, we did a full glycomic analysis, including IgG glycomic analysis in a large number of people living with HIV who are in long-term ART. They had to be on ART for at least 5 years and well-matched control. And indeed, we found that despite being in long-term ART, people living with HIV have an acceleration in the degradation of anti-inflammatory glycan and accumulation of pro-inflammatory one. So I'm showing you here an example of, again, what I showed you before, a glycan without the sugar galactose. And you can see here, both women and men living with HIV have an acceleration or increase in this pro-inflammatory, pro-aging glycans compared to their very well-matched control, again, despite being on antiretroviral therapy for 5 years with very high CD4 counts. The median here was 750. But this is just one example of a glycan structure that its level was altered in people living with HIV compared to control. But there were many other glycan structures that their level were altered, that we could actually take several of these glycan structures and put them together in a machine learning model that can predict the biological age of a person based on this glycan and compare it to chronological age, which is what you see here. What you see here is the difference between this biological age predicted by glycans and chronological age. And in black, you see this for people living with HIV, and you can notice the average is 0. You know, some people have some accelerated biological age, some people have decelerated biological age, but it's eventually averaging 0. While people living with HIV here in red have a clear acceleration in biological age of about 7 years based on the glycan. Again, taking a long story short, because this was already published, we did indeed found that this glycomic degradation correlates very strongly with inflammation, and also is more pronounced in people living with HIV who also living with an age-associated disease. But something we noticed while we're doing this study that caught our attention, and is something we're following up upon, is we noted that this glycomic degradation start to happen many years before people living with HIV experience this age-associated disease, suggesting that this might be a good candidate for a biomarker predicting the development or onset of this age-associated disease. So I give you an example here again of this study where we analyzed sample from people living with HIV on long-term ART who were either cases, and the cases here were people eventually develop GI-related cancer, an inflammation-associated cancer, or the control, which were people living with HIV who did not develop any cancer as far as we know. And we were fortunate that as we had access to sample from these two groups that were collected 5 to 10 years before the cases developed the cancer. And interestingly, 5 to 10 years before they even develop the cancer, we can see signs of glycomic degradation in them more than the control. As you can see here in the right, the people who will eventually get cancer 5 to 10 years later have more accumulation of degraded glycans, one again without this important sugar galactose or non-galactosylated glycan. And on the other side, the people who remain control have more intact glycan with more sialic acid and galactose and whatnot. So we thought back then, if this glycomic biomarker of aging can predict complicated disease outcome like cancer, can they also be measured before stopping therapy and predict time and probability of viral rebound? So we went back to this cohort of 246 people I mentioned to you, and we did a full glycomic analysis, including IgG glycomic analysis to examine if this glycan before stopping therapy can predict whether a person will become post-treatment controller or not, or how long will it take for the virus to rebound. And the answer was absolutely yes. Here, for an example, I show you several glycan structure that was measured in the blood of people living with HIV before they stop therapy and can differentiate who will become post-treatment controller from non-controller. And I mentioned to you the definition of post-treatment controller happened at least 6 months after stopping therapy, so it's something in the far future. And you can see here in blue, several glycan structure were enriched in the blood of people living with HIV compared to post-treatment controller compared to non-controller, and vice versa. Many glycan structure here in red, their level were reduced in the people who will become post-treatment controller, or in other words, they were enriched in the people who will not be a controller. And this was quite a strong signature that was significant and remains significant after adjusting for multiple comparison. And if I just want to show you one example of this glycan here, this glycan structure, for an example, before stopping therapy, you can see it's almost not existence in the blood of people living with HIV, but it's highly detectable in the people living with HIV who will become non-controller. And if we take this glycan structure and put it in a predictive model, you can see it behaves by itself very well, give you very nice area under the curve, very high specificity and sensitivity, and this is just one structure. Now we are combining this structure together in a multivariable model, hoping it can even enhance this predictive capacity. And this here is binary. We're looking at whether the person will become a controller or not, but we also found several glycan structure that is their level before stopping therapy can predict time to viral rebound as a continuous measure, which is showing here in this Cox proportional hazard model, where you can see several glycan, their level before stopping therapy can predict either a faster or delayed time to viral rebound as a continuous measure. So yes, these glycan are a potential candidate for a biomarker of viral rebound. But our next important question is this just a mere correlation, or they might have a functional significance in the phenotype? And we do actually have several lines of evidence that is these glycomic degradation might not just be a biomarker, but they actually can contribute to immune dysfunction during and after ATI. But in this one slide, I will just show you an example of this line of evidence. And as many of you know, the glycans on the Fc portion of antibody, which is what we measure, play a very important role in the ability of this antibody to bind to the Fc receptors on the surface of many innate immune cells, which will dictate the ability of these innate immune cells to do several antiviral mechanism or function. For an example, it can dictate the ability of the antibody to bind to natural killer cells to elicit what is called antibody-dependent cytotoxicity, or ADCC. It also can dictate the ability of the antibody to bind to phagocytic cells to elicit what is called antibody-dependent phagocytosis, or complement, for complement deposition, ADCD. Now, these functions are important for controlling virally infected cells, and we wondered if this glycomic profile or glycomic alteration compromises the ability of HIV-specific antibody to enhance these Fc-mediated innate immune function to control virally infected cells. So in order for us to test that, we took an HIV-specific antibody, in this case 10-1074, it's famous BNAB being used in several clinical trials, and we did a glycomic analysis on it, and inherently this 10-1074 wild type that is being given to people is having this glycomic profile called G0F. G0 means it does not have galactose, which is similar to what we see in people living with HIV with accelerated biological aging, and also people who will have a faster time to viral rebound. And F means it does have this other important sugar called fructose, which is very important for Fc-mediated innate immune function. So we took this antibody, we removed this glycan, and we did a glycoengineering to change this glycan. So we made this other glycoform called G2, which is G2 means it does have a lot of galactose. So basically 10^24 does not have any galactose. We added a lot of galactose. It does have fructose. We remove it to make this G2 glycoform, which happened to be the glycoform that is correlating with delayed time to viral rebound. So we have the glycoform correlating with a faster rebound here in black, the one correlating with delayed rebound here in red, and we take this same antibody with different glycans and we test it for its ability to enhance our innate immune system to elicit ADCC, ADCB, ADCD. And here for an example, the ADCC. And you can see in the x-axis, antibody concentration of isotype control, the G0F glycoform or the G2 glycoform. And in the y-axis is cytotoxicity, and you can clearly see that is the wild-type 10^74 is very poor in eliciting ADCC. It's about 17%. By just changing 2 sugars, you can dramatically enhance the ability of this antibody to elicit ADCC, going to almost 80%. Same thing for ADCB, same thing for ADCD. And I think this is very important for at least 2 reasons. First, it actually correlates with our in vivo data and can maybe explaining it. It's showing that this is maybe not just a mere correlation and they do have a functional significance. But the other reason, it's a very good example of what glycans are. This is the exact same protein. By just removing 1 sugar and adding 1 sugar, you can dramatically enhance its antiviral activity, which I think could have a lot of implications in HIV curative strategy that rely on BNAP to do their job, because again here by just manipulating 2 sugar you can dramatically enhance the antiviral activity of these BNAP. So again, this is just one example of how these glycans could be not just a correlation, but they could have a functional significance. But the other example I wanted to show you, as I mentioned, beyond the circulating glycans, how about cell surface glycans? And in order for me to show you this example, I need to introduce to you or remind you, if you already know that, this concept of glycoimmune negative checkpoints. And I'm sure all of you are quite familiar with classic immune negative checkpoints that works by protein-protein interactions, like the famous protein BD1 on CD8 T cells, adaptive immune cells, bind to this other famous protein BDL1 on pathogenic cells like cancer cells. And this binding sends an inhibitory signal to the CD8 T cells because BD1 carry an inhibitory domain, it's an ITIM domain, and that mechanism allows cancer cells, for an example, to evade immune surveillance. So that's in adaptive immune cells, CD8 T cells. What about innate immune cells? Are those left without any immune negative checkpoints? Of course not, because those are quite important cells in attacking other cells, and they have to be regulated under the same, checks and balances umbrella of our immune cells. But so the innate immune cells do express many immune negative checkpoints protein, but some of those proteins expressed in innate immune cells transmit their inhibitory signal by, instead of binding to another sugar, like BD1 bind to BDL1, they bind to glycan, sugars, and hence the name glycoimmune negative checkpoints. There are many of those in the surface of innate immune cells, but today, or in this example, I want to focus on those family of protein called Siglix. It's a family of about 15 proteins that express very highly in myeloid cells like monocyte, neutrophil, macrophages, microglia, but also a couple of them express in NK cells, Siglix-7 and Siglix-9. And Siglix are protein bind to the sugar called sialic acid, represented here by a purple diamond. And when you have multiple sialic acid together in a glycan structure, this glycan structure now called sialoglycans. So it has been shown many, many times over the last few years that these cancer cells cover themselves with sialoglycan. So, and the reason why they do that is because these sialoglycans in cancer cells allows them to bind to several of those Siglix, and some of those Siglix, many of them actually carry an identical ITIM domain, an inhibitory domain, identical to the one carried by BD1, CTLA4, TIM3, TIGIT, your usual immune negative checkpoints. So when this sialoglycan in cancer cells bind to the Siglix, that send an inhibitory signal to these innate immune cells and allows these cancer cells to evade immune surveillance. So this is like a very potent mechanism that is cancer cells use to fool our immune system by literally sugarcoat itself. And there is many beautiful examples on how this is a very potent mechanism in cancer immune evasion. But if you want to read more about it, I will refer to you to this study we published recently in Cancer Immunology, Cancer Research, where Pratima Sini, a postdoc in our lab, described how this is quite potent in allowing pancreatic cancer cells, which is one of the cancers that have a very strong immunosuppressive effect, to evade immune surveillance and how we can overcome that. So why I'm mentioning this? The reason why I'm mentioning this is because a soluble version of these Siglix, which is sometimes they can shed from the immune cells to the plasma, correlated with a faster time to viral rebound, several of them. So these glycoimmune negative checkpoints before stopping therapy correlating with a faster time to viral rebound, which was quite interesting for us, because while we were seeing that, this study came from here, from Spain, from Barcelona, from the group of RC-Kisia, describing the same phenomena. This protein, Siglix-3, which is also known as CD33, also its level in the plasma correlated with worse disease outcome after stopping therapy. So we thought, okay, this is consistent result apparently, but what it means, why the higher level of these glycoimmune negative checkpoints will correlate with worse disease outcome. Is that possible? That is, HIV infected cells use a very similar mechanism to cancer cells to evade innate immune surveillance. Do they also sugar coat themselves with the sialoglycan in order to bind to some of those Siglix and evade immune surveillance? And this question started this project in our lab led by Cellini and Shamsul, which is actually just got accepted recently for publication in Nature Communication. And this is a screenshot here from the proof. So hopefully it will be out in a week or two. And obviously for the time, I will not go through this paper in detail, but I will give you a summary in a nutshell where we indeed found that is HIV infection remodels the glycosylation machinery of its infected cells to do many changes, including increasing dramatically the expression of several sialoglycans. Again, it sugar coat itself with these sialoglycans. And we did a lot of work in this paper to identify which kind of Siglix bind to the sialoglycan, because not every sialoglycan bind to every Siglix. So we did a lot of work and found that is this sugar coat of HIV infected cells in particular bind to Siglix-3, again CD33, like the paper from RC-Kisia, Siglix-7 and Siglix-9. And those Siglix expressed in NK cells and myeloid cells, and we did a lot of work, mechanistic work in this paper using CRISPR-Cas and whatnot to find that is this binding send an inhibitory signal to this innate immune cells and allow HIV-infected cells to evade immune surveillance. So again, that is just an example of how we can use this biomarker data to identify a novel immune evasion mechanism HIV-infected use to evade immune surveillance. But the important question here was, can we stop that? Can we remove this, you can say, sugar cloth of HIV-infected cells? And we thought maybe we can use this enzyme called sialidase. So sialidase is an enzyme that can remove this excess sialic acid from HIV-infected cells, which will enhance their susceptibility for innate immune surveillance. However, sialidase will go and also remove this same sugar, sialoglycan, from HIV-uninfected cells, the healthy cells, which will make immune system also attack them. So we thought maybe we can conjugate this sialidase, represented here by this Bickman shape, eating sialic acid from the surface of HIV-infected cells, to an HIV-specific antibody, to a BNAb, with the idea here that is the BNAb will steer the sialidase to the surface of HIV-infected cells, remove this excess sialic acid from it, makes the innate immune system now do not bind to Siglix, makes the innate immune system attack it without impacting HIV-uninfected cells. So we went ahead and made several of these BNAb sialidase conjugate, and we first tested whether it selectively removes sialic acid from the surface of HIV-infected cells. And you can see here an example where we combined HIV-infected and uninfected cells in the same tube. We treated them with the antibody alone or antibody conjugated to sialidase. And then in the y-axis here, we stain them for sialic acid. And you can see nicely that is the conjugate removes sialic acid specifically from HIV-infected cells and barely touch HIV-uninfected cells. So the next important question here, whether this selective removal of sialic acid from HIV-infected cells enhance their susceptibility for innate immune clearance. And as you can imagine, we have tested this 6 ways to Sunday. In this slide, which is my last slide before the conclusion, I show you an example of this testing where we again combined HIV-infected and uninfected cells in the same tube, but after staining them for different dye. So the HIV uninfected with a red dye, HIV infected with a green dye. And you can see this here in this live imaging staining, but you also see the number of the cells here. We can add this to this co-culture natural killer cells, which is again an example of a cell that do have Siglec 7 and Siglec 9. Two of the Siglec we found to bind to HIV-infected cells strongly. And you can see the NK cells obviously will recognize HIV-infected cells and will reduce it by about twofold. But it's obviously far from being capable of removing all HIV-infected cells. You still see a lot of green cells. We can add an isotype control to this mixture and nothing happened. The twofold remain twofold. We can add two different concentrations of this BNAb. And the twofold reduction of HIV-infected cells now become 5-6-fold, because in addition to the direct cytotoxicity by NK cells, the BNAb will help NK cells to elicit ADCC, which will kill more HIV-infected cells. And you can clearly see the number of HIV-infected cells, the green one is reducing as you're going from left to right. And you will wonder if the Salidase conjugate can do better. And indeed, using the same concentration of the BNAb, but now adding Thylidase to it, this 5-6-fold become 25-50-fold, and almost all of the HIV-infected cells were wiped out from this co-culture. And very importantly, the red one, the HIV uninfected one, remained unimpacted much. This is just one example of one donor, but you can see this is reproducible using NK from multiple donors living with HIV. In this graph, you can see in the x-axis antibody concentration, whether it's conjugated or not to Salidase. And then in the y-axis is cytotoxicity. And you can see every time you add Salidase to the BNAb, you're increasing their ability to help NK cells to do cytotoxicity a lot. Now, NK cells have Siglec 7 and Siglec 9. Monocyte have Siglec 7 and Siglec 9. In top of that, also Siglec 3, which is very important. And indeed, when we repeated this cytotoxicity assay using monocyte as an effector cells, we can see even a more clear difference between the antibody alone and antibody conjugated to Salidase when we look at monocyte-derived phagocytosis. We also repeated this using neutrophil, which is usually do not contribute to the killing of HIV-infected cells, but by just adding Salidase, now they are, contributing to this killing party. We have tested this using autologous assay, but we also recently tested this in a humanized mice. And you can see here that when we added the Salidase to 10^10^74, the reduction in viral load was much more significant compared to the 10^10^74 by itself, which was not capable of reducing viral load. So just to go to my conclusion, I give you two examples today. In the first example, I showed you that people living with HIV despite long-term ART experience a glycemic degradation, and this glycemic degradation can be a good candidate for a biomarker for both comorbidities in people living with HIV as well as viral rebound. In the second example, I showed you that is HIV remodeled the glycosylation machinery of its infected cells to create a glycan shield that's protecting it from being cleared by innate immune cells. But really, those are just two examples of how we can utilize this multi-omic data from people living with HIV with documented viral rebound kinetics in order to identify what we think physiologically relevant pathway, because those are pathway that is evident in people with a post-treatment controller versus not, in order to develop what we call biomarker-driven intervention. Today, I just show you just two examples of a glyco-engineered antibodies, BNAb, or a BNAb conjugated to Salidase, and I showed you how these modification could dramatically enhance the functions of this BNAb. But again, this is just one omic, the glycomic. We have similar data for the proteomics and metabolomic data, and as I mentioned, we are learning more and more as we go with this project on viral rebound and HIV reservoir. So with that, I go to my acknowledgement slide, and first and foremost, thanking all of the volunteer participants in this study, the 246 people from 5 different countries, 15 different clinical trials. That is their sample with the essence of these studies. I would like to thank folks in our lab that I tried to highlight along the presentation, our funders and our collaborator, and thank you.
Pues con estas dos presentaciones creo que tenemos la forma fantástica de empezar una tarde-noche estupenda en Granada, que tengáis muy buena velada, que disfrutéis de la ciudad, de la comida y nos vemos mañana puntualmente por la mañana. Gracias.